Good morning, everyone. So I'm Florence Fombertas, Head of Marketing and Communication at Oncodesign, and I'm very pleased to welcome you today to our webinar entitled, How Do We Keep You in the Race of COVID-19? During the coming hour, our panelists will go through the panorama of infectious disease, the challenges pharmas and biotech have to face, and how our innovative solution providers, Allianz, bring complementary and integrated spectrum of expertise. This outstanding webinar will be animated by Yukon Tamin from Symbiose, CEO of Symbiose, Fabrice Viviani, Senior Executive of IP Oncodesign, Patrick Larcier, Senior Director of Drug Development and Regulatory from Pharmalex ARC, and at last but not least, Marie-Laure Sola, Director of Client Service and Scientific Expertise.
Sorry.
You get the floor is yours.
Thank you. Hello, everyone, and thank you, Florence, for this introduction. So, as mentioned, we will give you an overview on how our innovative solution consortium can support your drug developments. And we will focus today more particularly on our dedicated offer for drug candidates targeting COVID-19. So it is a perfect illustration of the ability of our companies to fit as closely as possible the needs of the market by offering our clients high added value research services in a constrained context linked to the urgency of developing therapeutic and prophylactic strategies against this virus. First of all, a few words and figures on the current epidemic worldwide context. Currently, as you know, 200 affected countries, more than 62 million confirmed cases, with just over 1.4 million deaths attributed to this infection. In France, it represents more than 2 million cases, including almost 52,000 deaths. In Europe, more than 18 million cases, including almost 350,000 deaths. And in the US, 15 million cases for more than 280,000 deaths. These numbers are unfortunately increasing day by day, as you know. Next one, please. So, paradoxically, pandemic outbreaks due to the emergence and spread of viruses or microorganisms that are pathogenic in humans are obviously not new, and the scientific community is sensitized and already prepared. Since its inception, humanity has faced different periods, epidemic outbreaks such as diarrhoeal, polio, plague, yellow fever. More recently, AIDS has been a viral emergence that has caused ravages in the human population. And until recently, the risks associated to the emergence of viruses in the human population with SARS-CoV-1, MERS, and H5N1 have mobilized and continue to mobilize the scientific community. What had finally remained at the pandemic infectious risk status at the time for SARS-CoV-1, MERS, and H5N1 ultimately resulted in a pandemic reality with SARS-CoV-2. It is indeed a respiratory virus that spread through the airway, apparently with slightly more severe pathogenicity characteristics, which is in connection with our lifestyles, has quickly spread throughout the planet. This virus has a considerable global impact that we are experiencing, ultimating having no means of protection other than those related to masks, gestures, and confinement. Next one, please. So it is this global impact that has led to the huge mobilization, especially of global research, aiming at developing therapeutic and prophylactic strategies against this plague. We are witnessing a completely new situation with massive and targeted investments in a single pathology estimated around $15 and $20 billion. The emergency since the first wave was to have access to a range of treatments already available, hence the numerous projects of repositioned molecules, which represents 40% of the products evaluated, in the context of more than 550 clinical trials that have been put in place in record time. This mobilization has generated a pipeline of molecules in development widely phased to date and evaluated to more than 1,300 products and vaccines, most of which are at the preclinical stage, and half of which are new treatments in development. And some of them, as you know, will soon hit the market. Despite these vaccines, which arouses real hope coming soon on the market, it's necessary to continue the efforts with the development of new vaccine and therapeutics. Next one, please. So it is clear that facing this huge pipeline and the emergency for available treatments, the offer of services and expertise to support the development of these products has obviously been organized with stakes at all levels. Preclinical, with the need to have the most relevant preclinical models to screen, assess the efficacy and toxicity of these candidates, and with the challenge of setting up reliable clinical trials in a very short time frame, which requires optimizing as much as possible regulatory constraints to go faster. So to give you a more precise idea of the ambitious challenges currently being faced in vaccine development for SARS-CoV-2, usually the development of a vaccine takes about 15 to 20 years. Today, the biopharma has succeeded to register the first vaccine within 8 to 10 months without deviating from all the steps of the development and authorization process. It's a genuine tour de force. So this obviously requires optimization at all levels, ranging from the technologies to design and produce the candidates, streamlining of preclinical and clinical developments, and agility and responsiveness of the regulators. This said, I'm pleased to turn the floor over to Patrick Larcier, who will tackle more the regulatory part of the development. Patrice, the floor is yours.
Thank you, Hugues. Next slide, please. So good morning, ladies and gentlemen. So we receive many questions about the timeframes, about the availability of treatments or vaccines against COVID-19. Many people pointed out that usually to bring a vaccine or drug on the market, a huge number of quality non-clinical and clinical studies are required, which takes between 8 to 10 years with a substantial financial effort. So how is it that we are talking about 15 to 18 months to bring vaccines against COVID-19 available to patients? Next slide, please. In fact, the competent authorities, in the circumstances of the health crisis that we are currently experiencing, have certainly taken special measures but have not put in place new tools to accelerate the development of this candidate for medicines or vaccines. Indeed, there are alternative regulatory pathways in Europe as well as in the US, but also in Japan for emergencies such as pandemics, threats due to infectious agents or pathogens. These paths are summarized in the slides you can see on the screen. In Europe, 2 initiatives are proposed by the European Medicine Agency, the EMA. PRIME was launched in 2016 to facilitate the development of drug candidates in pathologies with unmet medical needs. The idea is to initiate an early dialogue between the EMA and the developers for promising products to facilitate their development and then, of course, patient access to these treatments. The adaptive pathway is a scientific concept to improve access to new treatment for patients, but applies to treatment for pathologies with significant medical needs and for which it's difficult to collect efficacy and safety data via the classical approaches, such as the conduct of large-scale clinical studies that would unnecessarily expose patients to a drug candidate for uncertain benefits. In the US, there are several options that can be granted by the FDA depending on the pathology for which your future treatment is intended, existing medical needs, as well as the data available and so on. If a breakthrough designation, if possible, it's interesting to look for a fast-track designation for early programs and then continue with a breakthrough designation after obtaining more robust clinical data to allow for closer communication and exchange with the FDA. Note that in Japan, for instance, there is also an accelerated program called the Sakigeki strategy since 2015. Finally, there are two particularly interesting marketing opportunities in Europe, such as a marketing authorization under conditional approval, which is valid for 1 year, re-evaluable and therefore potentially renewable, obtained when efficacy and tolerance data for drug candidates are not yet available. Marketing authorization under exceptional circumstances is of interest as well, when it's difficult to predict the efficacy and safety of a product concerned in pathologies in which it's difficult to carry out some clinical studies, such as the Ebola virus, for instance. Facing the sanitary crisis, the EMA has set up a COVID-19 task force which aims to coordinate and enable the acceleration of the development of treatments and or vaccines against COVID-19. In the US, there are emergency use authorizations that do not correspond to a genuine marketing authorization, but makes these treatments available as long as the emergency situation continues. Next slide, please. As described on this table, some products have benefited from the various accelerated programs mentioned above. As part of the COVID-19 pandemic, Gilead Remdesivir was the first and only agent fully FDA-approved COVID-19 treatments in the US on the basis of 3 randomized clinical studies. In early November, the FDA issued several emergency use authorizations, first for the Eli Lilly monoclonal antibody bamlanivimab, and of course for the Eli Lilly drugs baricitinib in combination with remdesivir. A few days ago, the FDA as well issued emergency use authorization for one of the experimental drugs that the US President Donald Trump received while he was battling COVID-19, Regeneron casirivimab, and imdevimab, to be administered together. In addition to these treatments, approximately 200 medical devices have had the status of emergency use authorizations in the US. To date, 11 vaccines programs are currently in development or late development stage, 4 being more advanced than the other ones with various efficacy claims. 94.5% for the Moderna vaccines, 92% for the Russian Sputnik V vaccines, 90% for the Pfizer-BioNTech vaccines, and 70% for the AstraZeneca Oxford vaccines. On December 2nd, the MHRA, the UK agency, granted an emergency use approval to the Pfizer-BioNTech vaccines, whereas the EMA said they will make their own decisions by December 29th at the latest. In the US, Pfizer-BioNTech on the 10th of December will present the clinical efficacy and safety data of their vaccine candidate to the US FDA, whereas Moderna will do the same on December 17th, leading to a lot of expectations from patients, healthcare professionals, and regulators. So while these few examples can serve as example of the context of the COVID-19 pandemic, so far we assume that some of these vaccines will be approved via the same regulatory pathway that several other treatments or vaccines granted, such as, for instance, the smallpox vaccines in the US. Other vaccines, for instance, such as the ones against the Ebola virus, whatever we are talking about the Merck's vaccines with a single dose or the Janssen Labs vaccines with a prime boost approach, we're able to benefit from an accelerated evaluation in Europe and approval of their file either under conditions or exceptional circumstances, especially. Next slide, please. So this different and recent development on the battle against COVID-19 highlighted the importance of a valuable preclinical and early clinical package to support a clinical and regulatory strategy, as illustrated in the various and previous examples. Setting fit-to-purpose models and an early dialogue with regulators will help in successfully moving forward drug development projects. As such, biopharma companies may benefit from our innovative solutions to accelerate entry to clinical stage and market access. Thank you. So I think the floor is yours again, Hughes.
And a straightforward presentation on the regulatory strategies that can be considered by the biopharma industry. So it's now my pleasure to present our Innovative Solution Providers Alliance, which support what we provide to the biopharma industry to boost the development of innovative or repositioned drug candidates in the race against COVID-19 in this crisis context. Next one, please. So this alliance groups 4 French companies, Oncodesign, URBC, Pharmalex, and Symbiose. Going back to the origin of our alliance, the idea and the observation are very simple. Offer solutions that are truly able to respond to the challenges faced by our sector. Our role is not just a juxtaposition of skills, but it's to bring collectively a higher level of expertise and capabilities to what we could offer individually. What are we claiming? The ability to grasp the issues of our industry at stake in the 3 aforementioned dimensions, the preclinics, translational studies to the clinics, and of course, the regulatory environment in which all fits in an integrated offer, more variable than standalone studies. Indeed, we propose complementary services, expertise, and solutions, either innovative ones or proven ones, which are based on our different technology platforms. On top of this, we have set up a single governance structure to carry out projects on behalf of third parties. Next one, please. So to be very concrete, what are our strengths and values? The first point is to keep the agility and flexibility of each of the companies while building a solid organization to answer in a timely and effective manner our sponsors' needs. The second point is to answer precisely and in an adapted and optimized way to the needs of our customers. This is what we call fit-to-purpose solutions. Another important point is to answer to the translational challenges faced by our sponsors, meaning we aim at selecting the best suited models and solutions to support early projects until the clinical phase. So in our alliance, we indeed offer these panels of expertise ranging from the discovery phase to the early clinical trials. Of course, we know that even if we have a broad range of expertise and know-how all together, which we will present to you in a very practical way right afterwards, we sometimes need to surround ourselves with complementary experts. And in this context, we already have established networks of both private and academic experts and resources that can support your project and enable you to identify the most relevant solutions for your early drug development. Last important point, which we have already briefly mentioned above, is the need for you to have a flexible and agile organization that answers precisely your needs. For this, you should know that we already have established and signed an agreement of cooperation and collaboration between our members. Last but not least, we are all driven by core values that are both human values, such as honesty, respect, responsibility, and trustness, and also by business values, patience, integrity, and transparency. Next one, please. So we established a simplified process based on a multi-party confidentiality agreement with a unique contact, a unique project leader supported by a task force of the different companies involved in the project, and a unique study report on invoicing points. Thus, when the time comes, we can start working collectively with you and provide you with the best solution to speed up the development of your drug candidates. Next one. Now, let's move on the presentation of the spectrum of expertise of our members. Each company will now be going through their areas of expertise regarding COVID-19 research more particularly. And we will start with Oncodesign, then Symbiose, followed by ELBC and Pharmalex. So Fabrice, the floor is yours.
Thanks a lot, Hugues, for the introduction. So hello, everybody. So really pleased to really share with you what we have put forward since the recent months about our preclinical research, I would say, efforts as well to provide new solutions to the outside world, I would say, because we've been exposed to many demands recently coming worldwide about testing, I would say, new drugs, testing repositioned drugs, and as well new biologicals and new methodology of vaccination, really to make the proof of concept in a preclinical setting. So this has been something that we have undertook recently, I say, just at the outbreak of the SARS-CoV-2, I would say, pandemia, really to think what can we put forward quickly, because it was really a sense of urgency of this particular sanitary conditions. And we've looked, I would say, how to do about, because our pharmacology expertise at Oncodesign in many different disease indications, including virology and including what Hugues was saying, with good partners like Symbiose and other academic partners, we set up a particular, I would say, continuum of efforts to go about and really try to address, I would say, solution rapidly and provide the answer as quick as possible. And you can imagine in some cases, this has been quite critical to set up that really within the, I would say, important safety aspect of what we are doing experimentally. So we have put, I would say, a continuum of a solution starting from the in vitro biochemical assays, I will touch base in a minute, cell-based assays and in vivo models, really to really set the scenes about testing, or say, question a different step about the maturity of the solution people were coming to us and try to integrate through our models. In biochemical assay, you can imagine this was very important really to understand, and thanks to what already has been known in the past from the SARS-CoV-1, I would say, epidemic, about what was known about the, I would say, cell entry of the virus with a particular important receptor, human ACE2 receptor, for which we knew about which type of cells to use. And this was really rapidly demonstrated the SARS-CoV-2 was also using that particular mode of entry. And we've set up all the biochemical assay to do binding to the ACE2 receptor. People try to really, I would say, overtake that and try to inhibit this interaction with the spike S protein of the SARS-CoV-2. And as well, we've done as well some more findings recently about what is necessary to prime the spike S protein to enter into the cells with the need of a particular protease called TMPRSS2, for which we have as well set up a protease assay, try to find inhibitors of that. And we have people asking for that particular type of study. So we set the scenes really in vitro as well to really interrogate at the beginning some real early stage mechanism or what can be interfering with the entry of the virus. And this is all set up. Then it comes about the translation into cell assays, and that we have as well put, I would say, quite a lot of emphasis about this. If you go to the next slide, you will see certainly as well what we have developed, I would say, in cell assay. I was mentioned before, So we have the capability to really monitor the infection of the virus into different type of cells, including human cells, but not only the infection and the cytopathogenic effect, but as well the immune response on some human cells, especially human lung cells, where I would say this is the major, let's say, target of SARS-CoV-2. So we have been able to put that, I would say, in a shape where we can as well demonstrate what was found, a particular compound in biochemical assay, really translate in cell-based assay, including several human type of cells. So this is very important, really, to make the scene about taking a decision to move forward into, I would say, in vivo model. And this is what we have very put a lot of emphasis. And you can imagine this requires a lot of, let's say, protection and BSL-3 level type of labs, which we have set up in a very quick manner and fashion to have those type of, let's say, rigorous environment to manipulate this virus really rigorously. So two type of things were important to be taken into account here. Again, based on what we knew before from SARS-CoV-1 infection, that we knew that some rodent models were completely sensitive to a non-modified virus as well, and as well non-modified rodent model. So we have been using then the golden Syrian hamster model that has been completely set up really to interrogate drug reposition, new therapies, and as well vaccination methodology that has currently been testing in our labs. And we have been, I would say, very, let's say, invested about what type of readouts was really has to be set up quickly. Aside from the clinical monitoring, I would say, assessment, the viral load in the lung, in the blood, the organ histology, the cytokine profiling. I'm not saying here cytokine storm, but really some specific cytokine that respond to the virus. We're really monitoring and as well some phenotype of human cells. And this is currently all done today in the golden Syrian hamster model. The next, I would say, upper level about this is as well the infection in syndromal risk macaque, and which is also a very important strain and species really to go about a more complex, I would say, investigation about the mechanism of action of drugs, because we are on top of all the different readouts I was mentioning. We are also now applying as well imaging about we say CT scan about the lesions that occur in the lungs that can be applied and monitor to macaque. So that's really an important, I would say, assets of particular readouts, really to understand the biology of this disease and how new drugs impact several aspects of these. On top of this as well, as you may know, some of the severely affected patients that survive the SARS-CoV-2 infection sometimes developed an acute respiratory distress syndrome, for which you have, for some percent of those patients, some fibrotic element that's really stay long and really perturbates their lung function for many years. And this is as well try to say how to prevent the fibrosis that occurs to that infection. Well, today there is no models really to recapitulate on one shot that particular phenomenon, but we have at Oncodesign, based on our previous, I would say, expertise in IPF type of pathologies, on pneumonial interstitial inflammation, we do have models that resemble what can be happening with the SARS-CoV-2 inflammation. And that's why as well, for those who wants to interrogate antifibrotic drugs, really to help those patients that really come from a severe, I would say, aspect of the infection, really to help them to recuperate better in terms of not developing fibrotic in the lung. So that's as well aspects that we have proposed in our offer. Aside from that, which is the preclinical stage, obviously we have the capability to monitor clinical sample trial by monitoring the cytokine storm if needed, and as well the viral load from particular samples. That's as well an offer that we have put forward. And really what's important and what has been mentioned by you is about really having knowing better what is your needs, what is your problem that you want to face today with your new drug and new approaches. And really it's about collectively with all the partners, try to find the best solution to that and really try to understand what makes sense and what can be translatable to human. And this is very important, really, that we accompany you, and we are very, I would say, flexible about it. And this is where today I want to stop today my presentation, but really I'll be happy to talk any questions afterward, or even discussion afterward, because there's a lot to say about those preclinical models, and we still need to understand better the biology. And as you say, even the vaccination is really a great hope. There will be need to really continue to do some fundamentals understanding of this disease for the future, because those coronaviruses are still there, and we need to invest much more biology understanding about what's exactly, and I think we know a little bit and not everything about it. So thank you again for listening, and I'll hand the floor to Hugo back again.
A few words on Symbiose now. Symbiose acts as a pure player specializing on non-human primate models. As you know, models that are relevant on translational models to evaluate therapeutic and prophylactic strategies that target especially the immune system, such as immunotherapies and vaccines. A particularly recognized expertise in the field of infectious diseases in connection with the expertise of the team, and also the different models of infection, in particular by respiratory viruses, such as RSV, metapneumoviruses, that we developed during the 12 previous years. A preclinical offer or exploratory studies of pharmacology, immunogenicity, and toxicology, in particular the toxicology of the pulmonary compartment, through the expertise provided by its subsidiary Symbiose Respiratory. This company indeed has the unique possibility of implementing studies of aerosolization of molecules in these highly relevant spaces, one of the gold standard route of administration considered to address therapies against these lung diseases. A dedicated preclinical offer focused on infectious diseases through also a preferred partnership established with the Virpat academic laboratory, headed by Professor Lina and the Dr. Manuel Rosa Calatrava. So this laboratory is a reference laboratory on respiratory viruses, such as influenza, and its very next platform, which addresses all the virological analysis and expertise side, and also the availability of in vitro models of infection with SARS-CoV-2 on human reconstructed epithelium. So Symbiose, since April 2020, has been working also very heavily to develop and characterize a SARS-CoV-2 non-human primate model. The model has been developed on the African green monkey, a relevant species for infectious diseases, and intranasal, intratracheal challenges with a French strain of the virus sequenced by the Virpat academic laboratory, as mentioned. So this 7 days model enables to quickly assess the efficacy of candidates on the viral phase of the infection. We are able to monitor the evolution of the infection with various samplings such as nasal, throat, rectal swabs, bronchoalveolar lavages, and blood sampling. The available readouts range from local viremia, viral titering, immune monitoring, histopathology, and even transcriptomics if required. So we also propose the same model on the cyno starting at the beginning of the next year with the same readouts and allowing us to face the huge demand, as mentioned, of efficacy studies for COVID-19. Next one, please. Next one, please. Thank you. So Symbiose offers a bespoke offer on services mainly addressed to biopharma industry, thanks to an animal facility allowing to perform studies in confined environment levels 2 and 3, and with genetic modified organisms also. a capacity that will increase in the next year and a half with the building of our own facility and labs. Regulatory and quality standards eligible to work with our industrial sponsors, since our company follows the reference system of the good laboratory practices. Our animal facility is also accredited ALAC, with a particular focus on the ethical aspect and animal welfare, especially with its mainly veterinarians and animal technicians. The scientific credibility of the company consolidated through the strategic choice made 2 years ago now around the creation of a regional network of excellence led by Symbiose with an international vocation and name Symbiome, which was a winner of an investment program supported by the state and the Région Auvergne-Rhône-Alpes, and focus on the development of services and research and development projects on microbiota and infectious diseases. And an offer of services delivered by a team driven by values in connection with the company on a human scale, growing with all the agility and flexibility needed. Our company brings its expertise to support projects by offering tailored solutions and access to a network of qualified academic and private partners with advanced capabilities also. So finally, a scientific expertise, the company's development motor, supported by a strong desire of the team to be as close as possible to the needs of the customers in the context of quality human relations, to provide outstanding solutions within a full transparency and confidence framework. So, Marie-Laure, the floor is yours to present now ERBC services and activities. Thank you.
Thank you, Eric. Hello, everyone. So my name is Marie-Laure Sola, and I'm the Client Service Director for ERBC France. I'm very proud to be part of this partnership and involved in this webinar. It's a really pleasure for me to meet you in this virtual way. Just as a quick introduction, ERBC was created in 2019, 1 year ago now, when the French CRO CERM acquired the toxicology activities of RTC in Italy. At the moment of the acquisition, the new company was renamed to ERBC, a new name, but nearly 100 years of combined experience in the toxicology and pharmacology fields. ERBC is now an European leader in non-clinical studies to offering all services from non-clinical proof of concept to market of any type of drug candidate or chemical compound. In other words, from late discovery to first in human to provide safety information to support clinical development of a product. After these short introductions, let's focus on the topics day. How may ERBC help you with all partners in the context of COVID-19 crisis before first in man? Our multidisciplinary team of experts will support you to select the appropriate guideline adapted to your project. For example, the ISO norm for a medical device, the relevant ICH guideline for a medicine, as example, the ICH M3R2 for a new chemical entity, ICH S6 for a biologics, or the WHO guideline recognized by ICH for a vaccine. All regulatory studies conducted at ERBC will be GLP compliant. We will work with you in collaboration with the partners to set up and define the experimental phase. We will use simple rules to define the preclinical studies. The first principle is to mimic what will be done in human. Use the same route of administration, define, follow what you will do in the clinical trial. The second principle is to determine the number of species and to choose the relevant species to be used, 1 or 2. In general, we use rodents, mice or rats, and non-rodent species, non-rodents such as dogs, mini pigs, rabbits, non-human primates such as cynomolgus macaque or marmoset. The first principle is to define the relevant biomarker based on the nature of a test item tested. What will you include in your studies to follow the toxicity. The fourth principle is to use for the preclinical package a well-defined batch of test items as close as possible as the batch you will use for the clinical, mandatory for the GLP status of the studies. Finally, we will select the in vivo and in vitro studies and the appropriate analytical methods to be included in the project. Let's take 3 examples to illustrate these rules. Next, please. The first example, a new chemical entity. Simple. According to ICH-M3R2, you will need to do genotoxicity studies, repeated toxicity studies in rodent and non-rodent species, including a TK evaluation, safety pharmacology, the core battery package following ICH S7A and S7B, Irwin test for the system nervous central system, whole body plethysmography for the respiratory function, a telemetry studies in non-rodent species for the cardiovascular, and ERG studies for the cardiovascular function. For the GLP compliance, the formulation analysis will have to be included and the bioanalysis for TK analysis. I will take as a second example a vaccine, less common until now for us. In this case, the WHO vaccine guideline and the EMEA guideline for evaluation of a new vaccine will be followed. The preclinical package will be slightly lighter. The toxicology studies are limited to a single species susceptible to develop an immune response to the pathogen, including the evaluation of a local tolerance. To define the number of injections, the animals will be dosed with at least the same number of injections as intended in human, but with only 2 to 3 weeks between 2 injections. The principle to have the same therapeutic scheme as in human is not respected for these specific cases. The number of groups could be also limited to 3 groups, including a control, a placebo group, an adjuvant control group, and a vaccine group. The analytical methods will enable to follow an antibody level by ELISA or a cellular immunity by ELISpot. We are used to work with Oncodesign to define the most adapted methods. The evaluation of some biomarkers is recommended, such as CRP analysis or cytokine profiling. Finally, the safety pharmacology could be included as an endpoint in the toxicology studies or will be evaluated as standalone studies as done for a new chemical entity. Finally, as a third and last example, I will take a biologics, following the ICH S6, such as a monoclonal antibody, for example. The selection of the species depends on the test item. The animal should express the target. In most of the cases, this kind of test item is tested on non-human primates, such as cynomolgus monkeys or marmosets. Depending on the test item, a single species may be accepted. As usual in toxicology, we will follow the principle of mimicking the therapeutic scheme. In addition to the bioanalysis for toxicokinetics, there will be other assays focused on immunotoxicity endpoints, such as anti-drug antibody, immunoprofiling, cytokine profiling. In first instance, safety pharmacology endpoint will be included in the toxicology studies. Depending on the findings, standalone studies may follow. Further to this example, a case-by-case approach should be adopted in case of repositioning of drugs based on data already available. Preclinical studies may be limited to bridging studies. For ATMP, Advanced Therapeutic Medical Product, we can contact us to define the studies to be conducted. Now you are ready to go to clinical trials and to the next regulatory steps with Patrick Larcier from Pharmalex.
Thank you, Marie-Laure. So Pharmalex, as a consulting company, is proposing fit-to-purpose solutions We named these services InnoFlex, which combine our extensive experience and expertise with our innovative technology solutions to allow our customers to realize significant benefits for the early phases of development. These InnoFlex services applies to our chemistry, manufacturing, and control support from early phases of development up to the writing of the CMC modules in future marketing authorization application or BLA and NDAs. Regulatory and development strategy, of course, in preparing, setting, I would say, any type of meetings with regulatory agencies, for instance, classically in Europe and in the US, but in any places when appropriate. Data management and statistics, of course, with innovative options regarding study designs for drug developments and companion diagnostic test developments. Medical writing services, from the writing of what we call the study core documents, such as the investigator brochure, the IMPD, the IND, or NA protocols, to module 2.4 and 2.7 in marketing authorization application, as well as in BLA and NDAs. Of note, we can also support the writing of integrated summary of safety, as well as integrated summary of efficacies for the US filing. And last but not least, regulatory and pharmacovigilance services for pre- and post-market stages of development or commercializations. While PharmaLex has a currently presence in 18 countries, we have also set up an extended and selected network of experts and partner companies in the information field, of course, and of note, one of the point to be highlighted here, how strong reputation is based on our client-driven and flexible organizations. Next slide, please. So, as a summary, we tentatively illustrate the complementarity of services during the value chain of the development of a drug candidate, from the discovery to the conduct of clinical studies, including the non-clinical stage. With Oncodesign able to support you from the discovery to IND CTA for innovative therapies. With Symbiose as, I would say, an exploratory pharmacology safety and proof of concept studies expert in translational non-human primates models. ERBC for providing you with advices in complete and in a timely manner preclinical regulatory studies. And last but not least, PharmaLex with its capabilities and expertise in regulatory affairs, compliance, and pharmacovigilance for all therapies and medical devices. This partnership shows our willingness to have a full service offer from early drug development stage with scientific, high value, and rigorous inputs for our customers. Of note, we remain available for any additional questions you may have, and we thank you for your attention. Thank you. Merci.
Well, thank you very much to all of our panelists for this brilliant webinar. So here now is the Q&A session. So I've got a few questions coming up throughout the session. So are you ready, panelists? Just to give the answers. So the first one is addressed to Patrick. Here is a question. I always have a little difficulty understanding the value of the various options for accelerating programs in the US. Why do you say you should start with a fast-track designation rather than a breakthrough designation? Sorry.
Thank you, Florence. Yes, you are quite right. In fact, it's important to ask yourself the questions, and especially about these two potential tools that have been offered in the US. It's really depending on the nature of the data you have regarding your product, the ones under development, and based on the assumptions of the unmet medical needs for the targeted pathology. In general, with phase 1 and phase 2 clinical data, this is the sort of information you collect, and this is required for moving forward to what we call the breakthrough designation. Whereas with less robust data, for instance, it's time to and appropriate to move forward to fast-track designation. So bear in mind that as well, if you want to apply for the breakthrough designation, you have to demonstrate the superiority of your treatment over existing treatments. So overall, this is why we advise to move forward first with fast-track designation and then request potentially the breakthrough designation at a later stage to the US FDA. But overall, the benefits of the two options are roughly pretty similar in terms of shortened review, for instance, usually 6 months instead of 10 months, and in a way to facilitate the communication with the US FDA. But my recommendation will be, of course, first to ask yourself the appropriate questions, and then, of course, work closely with a consulting company strong enough in regulatory affairs, especially in the US, to advise you on these different topics. Thank you.
Thank you, Patrick, for this consistent reply. I've got another one regarding toxicology, and it's more addressed to Marie-Laure. What about the inclusion of safety pharmacology and toxicology studies, also for new chemical entities?
Yeah, thank you. The main issues is the dose selection, because if the doses are too high, a pharmacological effect may be hidden by a toxicological effect. If there is a too strong toxicity, the pharmacological effect will be very hidden. So we strongly recommend to do standalone study for a new chemical entity.
Okay, thank you very much, Marie-Laure, for your reply. I've got a question, another question coming up. Some vaccines are already in phase 3 and have been developed in the emergency situation that we know. Thank you for the presentation of your in vitro, in vivo services. But which animal models have been selected to assess the efficacy of these vaccines? And it is more addressed to Symbiose. So, Hugh?
Yes. So so we can say that definitively the first models used to assess the efficacy of the vaccine that's coming soon on the market, but also the immune response, were non-human primates. So it's very simple because they are susceptible to the same human strain of SARS-CoV-2. It's generally there are models reliable to study infectious diseases. So they are susceptible with the same strain, but there are a lot of similarities of the immune response, of the quality of the immune response. And another point that is we have a lot of tools to monitor the immune response in this species. So it's the reason why this species was very quickly used, and there were a lot of efforts to set up these models during the first wave of the infection. But in the second step, as mentioned by Fabrice, other models were set up on the rodents, and especially the Syrian hamster. And in combination with the non-human primates, they allow to screen, to assess the efficacy of the different candidates, and before to go to the non-human primates with an identified candidates susceptible to go to a human clinical trial.
Okay, thank you very much.
To add some
yeah, sure, go on.
Detail, I don't know.
Yeah, you can carry on. Yeah, if you want to add more details, please go.
No, thanks, Eric. We are completely, I would say, aligned with that. I mean, definitely the two models really makes the sense in terms of how to really assess the quality of the response of vaccination and these two models. For sure, the non-human primate is the most resembling to the human situation, but you can see the value having, I would say, a smaller rodent model really to make, I would say, faster evaluation because of probably of the drug quantity that you need to use at this stage when you are the exploratory phase. So really, it makes the sense about also capturing the information and the response in some I would say rodent model before really completing and have a more in-depth evaluation of about the response and the possibility of translate that into human in the non-human primate model, like the one that Hugues put forward. So really, it's a really continuum about where you are about the maturity of the compound, maturity of the mechanism actions you want to go afterward. So really, the two really are really well adapted for that.
Okay, thank you, Fabrice, for completing the information. A question regarding then again the in vivo models, and there's more addressed to you, Fabrice. Now, I mean, how do you qualify the hamster model of the infection by SARS-CoV-2 in terms of severity of the disease? And then the second part of the question: are you planning to develop models for more representative of the severe form?
Well, thanks for the question. Okay, so I said that currently in the sensitive species, that non-human primate and the hamster model, we have a moderate form of the disease. We're going to go to the death, I would say, which is the severe part of the disease that we will be able to, I would say, try to interrogate if we had that type of model. At the present time, we are in the moderate phase, which is most of the time what's happened to the human, because people really get along with the disease and again, I would say, get cured. Hopefully by themselves, there is a resolution, or you need some, I would say, therapies to really to help that. So, but by the fact that the hamster model is a moderate model, we do see a lot of traits about the human disease in terms of really making the value of the therapeutic translation. We do see, I would say, airway and lung inflammation with diffuse alveolar damage. We do see viral antigen in bronchial epithelial cells, pneumocytes, and alveolar macrophages. We do see body weight loss during the first days of the infection, and that's probably linked to the fevers that those animals get after infection. So we have the lymphoid atrophy associated with the mark, innate immune cells activation. We do see a lot of traits really where you have novel mechanism of action really to see what's the impact on those traits. So, and one aspect of the beauty of this model, which we try now to set up with specific, I would say, development we're going to try to do in the near future, is about because you have an effective transmission between the rodent animal. So really you can assess prevention therapy as well in this model, and we're going to set that in a couple of months to really make the case about prevention therapy. You've heard about some nasal spray really to try to prevent that infection and transmission. In human, but we will try to, first of all, demonstrate those concepts in the hamster model because this is possible. So technically, it's about developing that. So that will be the next outcome of this particular model. Now, going back to the second question about do we plan to make a severe model? There are severe models already available, and this is again based on the former knowledge about SARS-CoV-1 infection by using the key 18 transgenic human ACE2 mouse model, which is available already, commercially available. And this is very strong, very, very potent, I would say, model in terms of severity, because you get death within 5 to 8 days after the infection. So this is a mouse model available for that, very severe. We haven't yet put that forward. It's available, but really here you need to have very, very strong mechanism of actions really to interfere with, because the death occurs quite rapidly. So it's very severe model quickly, but it's available. We are abouting as well to put that forward. If there are, I would say, good, I would say, mechanism of action you want to try about, because definitely in terms of ethics, you need to have a good rationale to test combination of drugs within such a, let's say, severe model. So that exists. It's transgenic animals. I haven't seen any native animal model yet to produce that after the SARS-CoV-2 infection.
May I just add some details on what Fabrice explained. So yes, as he mentioned, the non-human primates are displaying not a very severe infection, just a replication of the virus in the respiratory tract. But so it's very, very reliable to assess, as I explained, vaccines or or therapies targeting the virus. But one way to develop, to increase the clinical signs in the monkey is to work on obese primates, because it's a real comorbidity parameters, as you know, in humans. And there are some research just starting, for instance, to set up these models, obese non-human primates infected with the virus, and to maybe improve the severe sign of the disease as we observe in humans. So it's one way also to improve and to have some models available to study the clinical side of the infection in humans.
Okay, thank you very much, Hugo, for this additional information that really shows the complementary of expertise between all the members. So I've got another one, and this is more addressed to Patrick. Is it possible to obtain the eligibility at the prime and the adaptive pathway in Europe for the same program?
Thank you, Florence. Yes, it's quite possible. In fact, these are two, I would say, regulatory approaches which are complementary because they are based on different concepts. Prime is intended to help you in navigating, I would say, and use the European regulatory universe. And once again, is helping you in developing your program. Whereas the adaptive pathway is more a scientific concept, and it's highlights in a way the level of evidence you have to put forward in order to obtain a marketing authorization and to maintain it. So very complementary approaches. But it's possible, of course, it's quite possible to, I would say, obtain in terms of during your drug development, I would say.
Okay, thank you very much, Patrick. I'm just having a look. I've got another questions coming up. Please do not hesitate. I mean, if you, dear audience, I mean, want to come up with questions before closing, and if I can see more questions coming up, please know that the webinar will be available on playback for the all biofeed session, and then available for the participants for the coming months, and we'll be happy, I mean, just to also answer directly to your questions. In the meantime, I've got a new question, and is again, I mean, regarding regulatory affairs. So, Patrick, if I take the recent example of Lilly bamlanivimab, do we know on which basis the FDA granted emergency use approval, for instance?
Thank you, Florence. Yes, of course, this is public information and it's available on the FDA website. I think it has been obtained through the conduct of 2 clinical studies, as far as I remember, which recruited about 500 patients in total. And based on part of this information, you could find information regarding the study design of 2 studies which have been conducted, as well as the primary and secondary endpoints which have been selected. And as far as I remember, for this program, I think that was mainly the fact that I would say, length of stay of hospitalization at I think 20 days or 28 days was a secondary endpoint, and that was the reason, one of the reasons, of course, of the approval of this product in the battle against COVID-19. As far as but everything is available if needed, and of course you can, I would say, gain further insights on the program.
Okay, thank you very much, Patrick.
You're welcome.
So as I said, I mean, here it is displayed on your screen. I mean, the contact information of each of the speakers. So please don't hesitate just to shoot screen or take the different email address. So again, thank you very much for all of you, dear panelists, and I hope, I mean, for the audience, you enjoyed, I mean, this very interesting webinar, and we wish you a very nice biofeed, and hope to speak to you very soon. So thank you very much, everyone, and have a nice day.
Nice day.
Thank you, everyone. Thank you for listening.
Bye-bye.
Thanks. Bye-bye.
Bye-bye.